For the next talk, I'm very happy to have Martin. We were just chatting away. Martin worked together with Johanna, actually, both come from the same lab in Graz. And so Martin is now going to extend on what Anna already showed us on gait-related brain dynamics, but he is using different approaches for source imaging, for example. And I'm very happy to have you here, Martin. So the stage is all yours.
Thank you. I'm very happy to be here too. And thank you for the opportunity to speak here. Very well introduced. Me and Johanna, we were working together, but afterwards she was going to San Diego, and now I'm here in Geneva for 4 years already. So somehow this is a small reunion today. I'm very happy about it, that we're part of this workshop, and then we were able to contribute something to the community, hopefully. Today I'm going to talk about investigating gait-related brain dynamics by EEG source imaging. Why I think studying gait or studying movement in general is important has two main reasons. The first one is more general. It is taking an active perspective on cognitive science, meaning we can have the perspective that cognition is not about creating a veridical model about the world, but rather about acting and interacting with the world, meaning we rather explore the world instead of passively perceive the world. And if you have this perspective on cognitive science, it already is quite obvious that movements are relevant and movements matter. And in general, we can start thinking about action and then trying to make a link to movement. And in that perspective, or in that framework, locomotion or gait is essential, because this is more or less the tool that we use to move in space to explore. And the second motivation comes from neuroscience, showing single trial activities or spontaneous neural dynamics recorded in rats here. And there are more and more works coming out showing if you analyze spontaneous neural activities, so these are single cell recordings here in a mouse model all over the brain. They had like thousands of neurons recorded. And what the authors show here, if you analyze spontaneous neural activities, what is predicting the neural activity most is behavior, meaning small movements that the mice are doing. Meaning what the authors are showing here, even in primary sensory regions, are mixed representations of the behavioral state and sensory information coming in. But that already indicates that there is nothing like, or there might be nothing like isolated sensory processing, but there is always some kind of a mixture integrating sensory information with the behavioral state or the movements these rodents were performing. And taking both together, I think that's already quite, I hope so, that this is a strong motivation in studying actions, movement, and gait. And that is somehow what I'm trying to talk about today. So I want to give you an outline of my talk. First, we're going to talk about mobile brain imaging of gait-related brain dynamics in humans and how we do that. I'm going to show you some results we achieved in the last years. And in the end, I want to talk about the future perspective to integrate subcortical signals in these analysis, because I'm going to discuss why I think subcortical activities are important when studying movements and gait. But first of all, let's focus on the approach I'm taking for mobile brain imaging. What we usually have is, when studying gait, is a person moving in space or moving on a treadmill, walking on a treadmill. And in order to study that behavior, it's important that we track the movements of the participants. And here there's a schematic I'm indicating, where there is a person walking in space, and we have all the joints tracked using motion tracking. And at the same time, and in synchrony with that recordings, we have high-density EEG to study the brain dynamics with it. And from these high-density EEG recordings, because they are just raw recordings, we have to do quite some advanced analysis to study what the brain is doing while we're walking. So in order to do that, we start with high-density EEG recordings, we could include the location of the electrodes, not to have only these recordings in time, but creating maps, and one map at a time, and then studying these maps. And then we can go a bit further using anatomical models. Let's say we can record the structure of the brain using T1 MRI scans and identify the gray matter volume, for instance, and using then this anatomical information to reconstruct brain dynamics from these scalp recordings. So the aim here is to include here anatomical and functional data in order to achieve brain imaging while people are moving in space. And one more important step or component in mobile brain imaging in gait or in mobile behavior in general is to take special care about artifacts. And here I plot one method I'm using or developing during my PhD. Usually when we don't correct for motion artifacts or muscle artifacts, we have quite a messy image. So if you don't do any corrections and you would just do this source reconstruction techniques here, you would end up with some brain activities that are spread over more or less all over the brain, and most prominently these huge red activities I'm indicating here are most strongly at lateral or anterior regions, but this is no physiological activity. This comes just from muscular activities. And it's a bit also a logical result, because we know the closer these brain areas are to the actual origin of these artifacts that are the muscles in the neck or the cheeks, the stronger would these artifactual source imaging will have some strong activities. So what you could do is trying to remove these artifactual activities. I'm going to talk about that a bit in more detail later, and try to remove all the artifacts and try to end up with much more nicer source reconstructions that are more distinct in space. But even if you did all these hard computations, then you end up with the brain images or brain dynamics that are vary in time. But that's only, I think, half of the job. I think the most important point or the most important component in mobile brain imaging is to link the brain activities to the behavior. Meaning, when we study brain activities and using complex imaging methods, we always end up with a blob highlighting up in the brain, right? But to somehow squeeze out the information that is relevant for the task, we have to link the behavior with the brain dynamics. And I think this is a very essential step in mobile brain imaging. So let me introduce how this distributed source imaging is done, meaning I'm going to talk about this first part here, how to come from the, how to translate this high density EEG recordings into time varying distributed source reconstructions. So what we can model, or what we can try to describe, is how, if there is a specific dipole active in the brain, as indicated with these arrows, and we call these sources J, how this would propagate from the brain through the skull to the skull, right? And one can do that, and we know how this is done from physics at about 100 years, sorry. We know that the electrical fields and the electrical potentials, they are spreading in a certain way, and this is very well described. And this is mathematically well defined as well. So what we have to do is describe the geometry of this propagation most. So what we have to describe here, or what we try to establish, is having a mathematical relationship between the sources and the scalp recordings. Because the only thing we record is on the scalp here, but what we want to know are the brain sources of these scalp recordings, right? So what we can do is make models. Here I'm indicating in A 4 different kinds of brains of 4 different individuals. You can see that they are varying quite in shape and size, and also cortical folding. And those are just MRI reconstructions of the cortical surface of these 4 individuals. And then you can try to co-register these brain surfaces with your actual scalp setup, meaning you can co-register the 3D electrode positions you were using for recording the EEG with your model of the head and the brain. And if you do that, this is already what I'm showing here. I'm showing here this brain surface co-registered with the electrode positions. And these geometrical relations are essential to describe how the fields are spreading from the brain to the skull, right? And if you have these models sorted out, the only ingredient that is missing is the connectivity values of the different tissues. But then if you assemble everything together, you can formulate that relationship in a linear relationship, meaning you describe, or you try to describe, the skull potential field as a linear function of the sources. And if you have that sorted out, you can also try to invert that. And that's a bit more challenging, because this is very well defined. There are no estimates, so to say, as long the parameters are fixed, you don't have to do any estimates. This is a well-defined problem. The ill-defined problem is to estimate the sources from the scalp, meaning you estimate the sources from your scalp recordings. And that you can do with different approaches. One approach that Johanna beautifully introduced and showed is dipole localization, that is heavily used in the community. Another approach is to use or focus on distributed source reconstructions. That is a different family of inversion methods. And I'm going to focus more on these distributed source models in my research. So that is just somehow should give you a general theoretical idea how I'm reconstructing sources from scalp recordings and why I'm showing brain images instead of scalp photographies in my talk. And what is very important while developing, so this is not, of course, not all developed by me. This has a history that goes back to 20 or 30 years. And this was very important and I think, yeah, hard to achieve method to have. And there are a lot of people working on this models for decades, basically. What is very important step, having done any estimate, is always a validation. So how can we validate that this is actually true, or that reconstructions provide plausible results? One way to, and I think also a very elegant way to validate these methods, is to have simultaneous recordings of non-invasive EEG plus intracranial EEG. What I'm showing here is epileptic spikes that are particularly strong. And those are just, this is an average of a lot of epileptic spikes. And you can see here the topography with it and different non-invasive reconstruction of the spikes. What I'm showing here below is different approaches to estimate the sources of these epileptic spikes, right? So this is minimum norm solution. This is the Loretta and Laura we're using in our lab. And you already can see that somehow they indicate, or they lead to similar results, right? But they're not identical. Meaning, depending on which inversion method you're using, you have slightly different results, yet the rough result is similar, right? It's always somewhere on the left parietal anterior sides, right? But I mean, we use these methods in our lab for about 20 years now to validate how to set the parameters and which models to choose. And for now, we're using the LoRa mostly, or S-Loretta in our lab. And what I'm showing here, that this localization beautifully matches up to the peak location in intracranial recordings, right? And this is how you can validate the methods, because you know where the electrical activity is maximum using invasive recordings, and then you can compare your non-invasive reconstructions with the ground truth, right? Does that make sense? So the point here is to highlight that these methods are also very well, not very well developed only, but also very well validated. So let's go back to motion science and studying gait and movements. Now I explain how I reconstruct brain sources from scalp recordings. When looking at, when studying movement, if you look in the textbook, what you mostly can see is primates moving their arms or doing central out tasks. And what was shown repetitively is that when this monkey is moving his arm, what we see in time frequency activities in the motor cortex. Here, I think the first plot comes from a primate, but we also can see in different modalities in ECOG, EEG, and MEG, there is always a very similar activity, meaning at moment zero, the monkey is starting to move, or the human is starting to move and doing this center out task. And what we see in all of these recordings using different species and different recording modalities is this very prominent suppression of beta rhythms that is indicated here in blue. So what I'm showing here is the time centered around zero, linked to the movement. And here this is the frequency on the y-axis, and in color-coded there is the magnitude of the activities. And there is quite some consistency between primates and humans, between LFPs, ECOG, EEG, and MEG. And that's very, very, very prominent features when we study LFPs or EEG signals while moving. So that's also what we did in our first gait study. Johanna, that was just talking before me, did a beautiful gait study in Graz, where we did our PhD together. And back at the time, it's almost, it's 9 years already. She did this experiment where she recorded 120 EEG electrodes while people were walking in a locomat gait orthosis. So this is not movement in space yet, but studying gait patterns. And this is how we started to study gait, basically. And with these 120 EEG electrodes, we also had the intraoral MRI, and also we recorded foot switches to have the gait cycle patterns. What I'm showing here is a time frequency activity logged to the gait cycle. One gait cycle here is defined as a right heel strike, a left heel strike at 50, and a right steel back hand. So a one gait cycle is one whole gait cycle from one right foot heel strike to the next right heel strike. And what I'm showing here is also this very similar to these previous results where people were studying hand movements or center out tasks, is this massive beta suppression here that is present over the whole gait cycle. Right? Can you see that? That's very similar to this beta suppression that was shown, I think, 30 years ago the first time. And it was very nice to see this feature, or to see this beta suppression, because we should see it, and we were expecting that activity to be here. And if you look at the sources, we could show that this beta suppression from all possible regions in the brain are located to central sensomotor areas, very close to where the legs are represented in the sensomotor cortex. So from that point of view, that makes sense, because that are the anatomical locations where the legs are represented in the sensomotor cortex. So what is matching is the cortical location of our source imaging, plus the spectral profile of the activity that we can be confident it was just like confirming that also during walking, when comparing to standing, we see this beta suppression, and they are locating to legs. The second feature we were focusing on, because this is all sustained, we just subtracted everything that is sustained there for the whole time of the gait cycle. And if you do that using the same data, we see these modulations in the gamma range, but those are occurring at a different frequency. Again, I show here a gait cycle from 0 until 100%. And in addition to this beta suppression that is visible, if you compare standing to walking, that is just a permanent suppression of these beta oscillations, you can see a more dynamic modulation in the low gamma range that is beautifully correlating with the gait cycle. And the novelty here is that those two features, they seem to be functionally different if you look at the frequency profile, right? I'm showing here a frequency profile where it's indicated that this beta suppression or event-related desynchronization in the beta range is maximum at around 18 Hertz, while these gamma modulations are maximal at around 30 Hertz. Right? I also show that here above. Here I show just the time courses of the gamma, and you don't see any, any sustained offset here. They're just almost modulating around 0. While the beta, if you just look at the beta time traces, they are suppressed and not as strong modulated. So meaning depending on the frequency you tune in, you have different phenomena you observe. And because there is this suggestion that different frequency spectra profiles indicate different functions, we were then moving on to try to observation if these two features are actually functionally different. That's what I did afterwards. So we were starting to sum up. We were starting with these classical features, looking at beta suppression and gamma modulation. And then I want to push everything a bit further, because when we go back, one very prominent feature in primates is this high gamma increase, right? That's visible at around 70 and higher frequencies. But classically in EEG, we don't look at these features. We rather go only until 50 Hertz, and everything above we consider as being artifacts. And I wanted to highlight the question, is that actually true? So I wanted to investigate not only these alpha and beta features, but also try to pose the question if we can observe something in higher frequencies that might be physiologically plausible. And for that, I introduced this artifact rejection method that is working with separating different spectral profiles. So as I indicated before, if you don't correct for any artifacts, you end up with very messy and blurry and highly contaminated activities. So the trick here is to separate different spectral profiles and always remove the strongest one, meaning the one that is strongest in power and also the one that is most spread in space. And if you do that, and that works for all the participants I was working on so far, if you do that, you can come from this, you can transform these raw source images to much nicer representations over here, where these obvious artifact contaminations on the side are just gone. And what we see here when we focus on high gamma range at around 70 Hertz is again this quite spatially distinct central sensory motor activity that is located very closely to where the legs are represented. And that's very nice. And in terms of time frequency activity, I was talking about this beta suppression already. What we can see when we go for higher frequencies is this very specific 70 Hertz high gamma increase that is also modulated. And that was already reported for primates or mice before, which is quite nice. So in summary, we find this beta suppression, high gamma increase, low gamma modulation, and high gamma modulation that is opposite to the lower one, right? So because we did this observation that the suppression and modulation features they seem to be functional different because they have different profiles, I was questioning myself, how can we show that, or how can we prove or give more empirical evidence that those features are functionally different. Sorry. No, let's first. I want to show you an example that this actually works and give you some idea what we can do in the lab. So I want to show here an example of these results, just focusing on this low gamma modulation for now. What I'm showing here is an experiment we did here not after my PhD here in Lausanne and Geneva. And we have a bit a more sophisticated setup now, where she's wearing an EEG cap that is more lightweight. There is an amplifier in the backpack. There are motion tracking sensors as well, non-invasive, wireless EMG markers. And if you assemble all this setup, what we can see here on the behavioral side, she's walking here on the treadmill. And from these motion markers, we can reconstruct the kinematics. And we can use these kinematics to identify the gait cycle periods, and then visualize the source images, as I showed you before, not only in a static way, but also in a dynamic way, meaning we have one source image per the time instant. And this is what I did. Here is the gait cycle indicated above. And this should show you these gamma modulations. And these are the sources, time varying over the whole gait cycle. What you can see here, the most prominent activities is, of course, on leg areas, but not only, there are also these parietal activities being active, but mostly there is this leg activities modulating correlated to the gait cycle. That is the main feature I was focusing in this study I did here. But to prove that this is no artifacts, because we were confronted with quite some skepticism when we did our PhD, and afterwards, when we're talking about the conferences, also if we talk about gait-related signals in the brain, there is always a justified question: is this artifacts or not? And how can we know it's not, right? So what I did here is two things. I tried to provide more evidence that these different features, meaning the beta suppression and the low gamma modulation, are functional difference. And second, I wanted to show that we also have these gait-related modulations if we just tap our fingers, right? And this is just much more controlled environment, because I said if we have these gait-related modulations here, we should also see that if we do a repetitive finger tapping task in a Faraday cage, meaning there were no cable swings, there were no environmental noise, and everything was just more controlled. And the hypothesis was that because we think the beta suppression and low gamma modulation are functionally different, they should be organized in different large-scale networks when we tap our fingers, right? Because the fingers are represented more lateralized, meaning the right hand is represented on the left hemisphere. And having this we can use this lateralization in trying to identify large-scale networks that might be different for the suppression versus modulation feature. Right? That is what I was focusing on a follow-up study. And I'm already showing the results here. This is what we observed in the gait study, where we had these modulations in the low gamma range, and very similarly, not identical. When we look at one phase of a tap, it's just really going with the finger once to the front and to the back. If you do similar analysis, we see very similar features. And again, we see, you can imagine this is a trajectory of your finger. So first there was no movement, and then there were like 9 taps over here. And what we observed here is, again, there is not much going on. And then this very strong suppression of beta oscillations indicated here in blue, very similar to the one I showed you before. But on top of those, so if you regress all these sustained effects out, we have these modulations coming up. And this is, you just look on the left or right hemisphere of your motor cortex, you already almost can count, these beta modulations, and then you already get almost a good idea how many finger taps you did. And that was quite nicely confirming our previous observation that that first is it's not artifacts, and second, those two features, meaning the suppression of the beta rhythms and the modulation of the gamma rhythms, are functionally different. And one more thing I want to highlight, this is also a bit it's also different in space, meaning if you look at this beta suppression, I'm showing here these brain images over here. The lower row is the beta suppression, or like power suppression and increase for different frequencies. And blue is always negative, and warm colors are always positive. And when we look at alpha, beta, and gamma rhythms, we see this suppression is very indicative of which limb we move, meaning we move the right hand or right fingers, while these modulation features are different, right? So if you compare in this beta 2 range, the functional networks or the sources of these features, meaning the gamma modulations versus the beta suppression, we can see different networks, different large-scale networks, right? This is even more lateralized to the ipsilateral hemisphere. And these static image, I also did a visualization here. meaning one step cycle, you can see that there is activity coming from the front towards motor areas. This is linked to one movement cycle in your tab. And what I wanted to show here above is this very first activity here. And this is just the indication of the experiment, meaning the experiment was the following, that people were instructed with visual cues what they should do afterwards. And these modulations over here, they were puzzling us in the beginning. And then I said, okay, I just try to get an idea what's going on here. And of course, there must be visual processing, meaning translating the visual information towards information that you can use to complete the tasks. But as you can see here, there was no movement performed over here. And this I'm showing here. First, there is visual information, so visual activity, and then motor activity. And that's very nice because there is first really visual processing and then motor processing, but there was no motion going on. Meaning if the instruction would be, an arrow on the screen means looking here and there, then we would not expect, I think, motor activity, but we do. And I think we only do because the instruction was a left arrow means the movement of your left thumb, right? So that already indicates that the instruction or the prospective task is influencing how we process visual information, because the relevance is to move in that task, right? So to sum up and to discuss these features, what we saw in my non-invasive reconstructions is this beta suppression indicated here in blue, and high gamma increase indicated here in yellow. For the gait, what was both in leg areas, while for the finger tapping, it was in hand areas. And that was very nicely consistent with previous mapping studies using electrocorticography, which was nice because there is no non-invasive modeling needed, because they are direct recordings, right? For instance, the hand is very beautifully represented here in this contralateral areas, what is beautifully matching with all non-invasive reconstructions, which gives us additional confidence. In addition, it is also consistent with these somatotopic representation of the motor cortex. This is a coronal slice of the motor cortex, where you can see here that the legs are represented here in the center, as we show it here, and the hands are represented more lateralized, as we show here. So these results make sense, and they roughly indicate that these desynchronization, synchronization, sustained patterns, they mostly represent which limb we are moving or which body part we are using. And to give a more functional interpretation about this beta desynchronization features, I show here a really ancient study by more or less or academic granddad that was founding the Graz BCI lab. And he did a study using isolated leg movements. And back then already, when looking at the CZ electrodes, that is just on top of these leg areas here, what was beautifully showed already over 20 years ago is that you have these myriad beta oscillations, but as soon as you're moving, those oscillations are vanishing. So then, if you then sum up and just look on the amplitude envelope, you can see not much is going on, and then this envelope goes down, right? So coming from the oscillations, we have the envelopes, and here this is just the color-coded version for all of possible frequencies, right? This was isolated leg movements, but it was very similar to our gait-related desynchronization patterns we observed in our gait study. And functionally also that makes sense because as Johanna already discussed, in Parkinsonian patients, there is hypersynchrony in the beta range, and this hypersynchrony in the beta range, it correlates with their problems in bradykinesia or rigidity. And when these Parkinsonian patients are treated with levodopa, what could be observed, meaning on and off medication, is indicated here in this spectrogram, that this hypersynchrony in the beta is vanishing, and this suppression of hypersynchrony in the beta is beautifully correlated with the improvement in rigidity problems. The more the lower the better, the more flexible you are in your movements. And that could indicate that if you're not able to suppress your beta rhythms in the motor cortex, you might lose somehow the computational flexibility in order to initiate your motor programs. And in the second way, looking at these high gamma features, again, on and off medication, what the author showed here that on medication, there is higher coherence between basal ganglia and motor cortical activities, especially at 70 Hertz, what matches nicely with our results. And taken both together, that led to the viewpoint that these lower frequencies, they are perceived as antikinetic, while the higher frequencies at higher gamma, we say 60 Hertz and above, are prokinetic, meaning beta oscillations, they so to say prevent movements in a very rough formulation, and high gamma activity are facilitated. So that was also shown by and confirmed by TAX experiments boosting either beta or gamma. On a second vein, to discuss this movement phase-related gamma modulations we showed for gait and also for finger tapping. That was replicated afterwards. So this is a replication I did in a single subject here in Lausanne. It's very preliminary results. That's why it's not as nice as the group results shown over here. But you can see the pattern is very similar. But this is treadmill walking, not locomat walking. And the day after tomorrow, we also will find these patterns, or we try to replicate these patterns using the Berlin data set in this workshop. Again, this pattern was replicated while looking on bicycling and walking by a different group, what is very important that somebody else is replicating our results, and also looking at other circular movements as circular arm movements here by Reinmar Kobler, showing these very nice 20 Hertz modulations that are correlated with circular hand movements that were really like large scale like this. And when looking on subcortical activities in the subthalamic nucleus using DBS electrodes for recording, there are again these gamma modulations in foot stepping that are quite similar to what we found. I think like taking all together, we can be quite confident that these modulations are physiological because other people were finding them, and they were also replicated with intracranial recordings, which gives us additional confidence. So I want to give you an intermediate summary, meaning that we can use the methods I showed you here for mobile brain imaging, or using creating a mobile neuroimaging tool based on EEG source imaging, spectral decomposition to suppress muscular artifacts, and then try to investigate gait and movement phase related activities. In that respect, we can separate different cortical systems that we suggest to be related to gait versus the movement state related networks that might up-regulate cortical excitability during movements, which is related to which limb is moved or which task to select. And the second ones are movement phase related networks, which might be related to the integration of sensomotor variable, but that we have to test a bit more over. Now I want to change gears a bit and try to go into the future perspective, or where I can see what we are missing and what we should, or what I was missing and what I want to focus on in the future. So when talking to our colleagues studying movements in rodent models, what is the most prominent area in the brain people are studying is usually the hippocampus or the entorhinal cortex, right? And this is an area we were completely not focusing on in our in our studies. But when we think about locomotion or movement in space in general, this is a very essential feature. Meaning what I've showed you before, or what I showed you so far, is gait-related signals that are related to the gait pattern. But I didn't focus at all at navigational signals, really. And I think if you wanna move from these treadmill-based studies more studying movement in space, so locomotion in the context of movement in space, we also have to focus on path integration and grid formation, as it was suggested by Budraki and Moser, and trying to make a link also to memory, right? Episodic versus semantic memory. And in Dover, it is essential to study and include subcortical signals as the hippocampus as well, because we know these navigational signals are strong and they are essential for moving in space. And I think this is a point I was completely missing in my previous research, because I was just focused on cortical signals, right? So what we could do is posing the question, is subcortical electrophysiological activity detectable with high-density EEG source imaging? Because as again, there are new studies investigating thalamic activities, hippocampal activities while people are moving in space. We're using intercranial recordings. And when I was seeing that, I was becoming very interested and thought, yeah, that is something we should include in our studies. But yet, this is very challenging because the textbook tells us in EEG we cannot see any subcortical electrophysiological activities. In the second vein, there is even a model suggested that the thalamus is essential for not only to relay information between different areas in the brain, but to adapt the connectivity patterns between or within a cortical patch when we process information. Taking both together, highlighting the relevance of the hippocampus plus highlighting the relevance of the thalamus, already highlights the importance in including subcortical signals when studying the brain while moving. So I was very happy in my lab to have the unique opportunity to record simultaneously from deep brain regions in the nucleus accumbens and in the thalamus, simultaneously with these intercranial recordings I'm showing here with high density, meaning 256 channels on the scalp. And if you analyze these signals here, if you derive from these 4 leads I'm showing you here, 3 bipolar derivations, the deeper you go into the structure, the stronger these oscillations get, meaning there are only low alpha oscillations on the very top, and the deeper you go, the stronger these oscillations get. And also on the time course, you can see that, if you go into the structures, what we record is very noisy, but if you're in the nucleus accumbens, actually we have these alpha oscillations that are much stronger in the structure than outside of the structure. And then we focused on analyzing the envelope of these alpha oscillations to compare the invasive recordings to the non-invasive. Meaning what we saw in the non-invasive scalp recordings is this strong alpha peak here, and this alpha peak was matching in frequency with the alpha peak in the PSD of the intercranial recordings. And then I looked at the alpha envelope of the EEG scalp recordings and the alpha envelope of these intracranial recordings, right? And what you can see already with your naked eye, looking at the structure over here, that these envelopes are very similar, right? I mean, the time range is quite big because I show here 100 seconds. But if you look at the rough envelope of these alpha signals, you already can see by naked eye, there might be some similarity, meaning there is, of course, some shared information. But to quantify that, and also to get an idea if you actually can detect these signals recorded with the intercranial recordings non-invasively, I did a study where I was taking again the high density recordings in combination with the 3D localization and to reconstruct sources. And then I used these purely non-invasive reconstructions to compare every point in the brain with these intercranial recordings, meaning what I did is just a very simple correlation between the envelope I was actually recording in these intercranial sites with the non-invasive reconstructions. And what we can show is that the correlation between these two is the highest and highly significant only at areas and sites that are close to the actual ground through implantation site, right? So what I'm showing here is a T2 scan with a overlay CT scan, and this blue dot is indicating the actual recording site of the intracranial recordings. And in these hot colors, I'm highlighting the correlation between the non-invasive reconstruction and the actual recordings. And that was also shown, that was also present in the nucleus accumbens recordings, where I even could show some slight lateralization. Again, here, these electrodes are showed in blue here, left and right electrode. And if you correlate with the left electrode, you have this bilateral pattern, while you correlate with this right electrode, you have slightly lateralized to the right side in the nucleus accumbens. And that was also present for the thalamic recordings, where the ground truth is here and the activity was mapped somewhere here. And in the third subject we had, we only found a significant activity for the left hemisphere. That is because on the right hemisphere, we did not see any alpha peak, meaning we did not find in the recordings, in the intracranial recordings, any physiological signals we could highlight or try to reconstruct. So if there is no physiological activity there, we also have a hard time reconstructing that. So from that point of view, that makes sense that it's just on the left, because on the right electrode, we could not find any meaningful physiological information. And just to look on the timing, these high significant correlations were just there for around 0 time lags, what must be the case, because as soon as we destroy the temporal relationship between the intracranial and non-invasive data, these correlations they vanish. Here I'm just showing across correlation plots, and this is highly significant for at about 0 lags, and for higher lags, this is just chance. And this is exactly how we investigate the statistical significance of those results. So to sum up, I want to discuss that we were able to show that non-invasive EEG is able to reconstruct some cortical signals. At the same time, our colleagues in Marseille were showing very similar results while focusing on SEEG recordings and simultaneous MEG recordings, here indicated in the thalamus again. And here they also had a hippocampal recording, which we did not have, meaning when we summarize these results, we can show that we can detect subcortical signals using high-density source imaging in the thalamus, as well as in the hippocampus. And taking all this together, meaning summarizing our study with the study of our colleagues, we can conclude that scalp EEG contains subcortical activity. It is possible to reconstruct subcortical activity from scalp EEG using source imaging, and the precision is limited by the method, meaning that there is an excellent time resolution, excellent temporal alignment, but moderate but usable spatial resolution. And if we now try to sum up all of these studies, meaning coming from gait-related or gait phase-related activities and focusing on cortical dynamics I try to introduce here, what I think is missing is these navigational signals, and then focusing on subcortical signals in order to integrate subcortical information to these core subcortical activities. Meaning what we want to have in the future is, of course, in identifying large-scale functional networks in the cortex, but also including subcortical signals, which we know are essential for spatial navigation. And with that future outlook, I want to close and thank my collaborators as well as funding agencies. And I want to thank you for your attention. I'm very happy to take some questions.